So this is chapter 8, section 4. For chapter 8, section 4, we are going to be looking at the central dogma of genetics, right? Which is that information goes from DNA to RNA to protein. It kind of flows in one direction, where the information on how to make proteins comes from RNA, and RNA, the structure of it is made from DNA. So if you look at this, DNA making copies of itself is called replication. So DNA makes a copy, makes a copy. That's replication, replication. When you go from DNA to a temporary copy like RNA, that's called transcription. The reason why it's called transcription is DNA is made with nucleic acids, RNA is made with nucleic acids. So they're both made with nucleic acids and therefore are linear. It's like the same language, right? If you make nucleic acids are both made with nucleotides, you keep the same alphabet, so you're just copying it over. That's what transcribe means, to copy it over. RNA to protein, however, you're going from nucleic acids written in nucleotides to proteins made in amino acids. So that is a translation. You're changing the language. You're going from nucleotides to amino acids. DNA does not have any other function except to be the source of information for proteins. Okay? It doesn't give the information to make proteins, it is the information to make proteins. So we need to look a little bit at RNA, this temporary copy. A few things about RNA. RNA has a sugar called ribose instead of deoxyribose in DNA. RNA uses uracil U instead of thymine T. And our RNA has a single strand instead of a double strand, which is why it's a temporary copy. Uh, and then this just says that transcription makes RNA from DNA. So here you have the DNA. You have to unwind it. If you remember, helicase is the enzyme that does it. Anything that ends in ASE, is an enzyme. So here it actually, this is helicase. It unwinds the double-stranded DNA, and then it makes RNA polymerase, goes ahead and makes an RNA strand from one side of this DNA. Unlike DNA polymerase, which makes DNA, RNA polymerase makes RNA. The DNA then winds back up again, and you have RNA, which is a temporary copy of the gene that would make the protein. That's what genes are, sections of DNA that make protein. Uh, to better understand this process, we need to understand the players involved. And RNA has three players. The messenger RNA, which has the message, the information to make the protein. Again, DNA is a structure that holds the information. It is the information. RNA is a copy. Messenger RNA is a copy of that information. Ribosomal RNA, which makes ribosomes, which is the part that's in charge of making proteins. I don't know if you remember, ribosomes are the part of the cell that make proteins, and it's an organelle. And ribosomes, ironically enough, surprisingly, have ribosomal RNA. Transfer RNA moves nucleotides, moves amino acids to the next, to the protein making area so that you get the next production going. You get the next amino acid into the link. And we'll show you how that works as well. So transcription and DNA replication. Remember, transcription is from DNA to RNA. Replication is DNA to DNA. There's a lot involved in it. We're really brushing over it with a very light brush. There's a lot of information to it. It's complicated, and it has to happen fast. So replication copies all of the DNA and makes a copy of it, whereas transcription does just one gene. So one gene will make one protein, and that protein will actually make whatever it is will make the protein you're trying to make. So here's your DNA. Okay, here's your RNA strands coming out. The main branch is DNA. The RNA is coming out from the main branch, and this section is a gene. So you're making copy after copy after copy of RNA of that gene. It looks like a leaf almost, right? Well, the main vein down the center is the DNA, and the sub veins coming out of the sides is the RNA. And that's just showing you how it would be made. And that's chapter 8, section 4.